Live.
Good evening, everyone. I would like to thank you all for taking out your precious time from your busy schedule. This webinar is organized by Banai, Behavioral and Neurosciences Academy of India. Banai is a not-for-profit organization formed in 2012 and has organized more than 200 plus programs nationally and internationally. Focus of Banai is to provide contemporary cutting edge on behavioral neuroscience. Banai is formed and run under the guidance and chairmanship of Dr. E. Mohandas. Banai welcomes everyone and anyone who is interested. It gives me immense pleasure to inform you that Dr. Bhaskar Mukherjee is the moderator for today's webinar. Thank you, Dr. Bhaskar, for accepting to be the moderator for the webinar today. I would now request you to take over and start the session proceedings. Over to you, Dr. Bhaskar.
Yes. Hello. Am I audible?
Yes.
Yes, sir.
Okay. Since yes, talking about the basics of multiscale neuroscience in the previous edition, in this edition we are back with demonstration of how multiscale neuroscience works if we take a specific symptom like schizophrenia or a specific symptom like depression. And Ram Bishnoi, our previous question speaker, is already there to elaborate more on this topic. Over to you, Ram.
Thank you, Dr. Bhaskar. So, can you share my slides now? Hello?
Yes, sir. You have to share it from your side.
I'm already sharing them now. Okay,
Yes.
is it good? Is it good?
Yes, sir.
All right. Well, welcome everybody once again. Talking of the neuroscience of psychiatric disorder. In last session, we discussed about how neuroscience is so fundamental to our understanding of psychiatric disorders. It's very important to understand how brain is organized and how it functions the way it does to understand the normal behavior and also in the disease processes. So today I'm going to focus on psychiatric disorder, specifically schizophrenia, to understand what we understand about what we know so far about the pathophysiology of schizophrenia from the multi-scale neuroscience. Before I go into specifics of schizophrenia, let me go through this slide one more time, which we discussed last time. And what I mean by multi-scale is that at micro scale, we can look at the brain cells and subcellular structures and processes like genes, proteins, neurotransmitters, etc. And at macro scale, we can look at the brain from neuroimaging point of view. We can look at the neural circuits and systems. And both these views at different scales are important. And even though they are codependent with each other, but it's important to understand them together so that we can make a sense of the whole brain functions. And it's also important to know how they different scale of the brain structures control the behavior and are implicated in mental disorders like schizophrenia. So this is how the temporal and spatial scales of organization of brain looks like. And if you see from the very small scale to large scale, there is a significant overlap between the scales. And what that tells us is that the neural signals which are coming from the different structures, say for example cell layers or dendrites or columns, they overlap, and uh, depending on the kind of modeling we use, uh, sometimes we are using the same neural signal and processing at different levels. But if we go through the small scale to large scale or macro scale and the findings in schizophrenia, we will discuss. I'm going to discuss that in a little bit, but we will understand a lot about how the whole system is influenced by the disease process like schizophrenia. So schizophrenia is a very complex neuropsychiatric syndrome with very heterogeneous presentation. It has a very complex genetic, neurobiology, and phenotypic profile. So it makes all the sense to understand schizophrenia from neuroscience point of view first, uh, because of all these issues. And then again, I mean, it's one of the challenging and serious and persistent mental illness we all come across in our clinical practice. So in 1890s, Kraepelin first described the syndrome, what we call now schizophrenia, and the Bleuler later termed it as schizophrenia. He recognized that it was different from manic depressive disorders, and he called it dementia precox because of the cognitive impairment associated with it. He was a colleague of Alzheimer and Huntington, and he was convinced that, like those neurodegenerative diseases, schizophrenia was also a disease of brain, and he spent a lot of time and energy looking at postmortem schizophrenia brains. Unfortunately, he could not discover any specific abnormality, and so do the several neuropathologists over next several decades. Uh, but what was noticeable from all neuropathological finding was that there was no gliosis or glial scars, which is a hallmark of neurodegenerative diseases like Alzheimer's disease or TBI. In the following years, then because of the absence of neuropathologies from the pathological observations and increasing influence of psychodynamic theorists, led to the psychosocial explanations of schizophrenia. And that's when the concepts of refrigerator mother and double bind came into use. And they were used to explain the cause for schizophrenia. Uh, Eventually, like neuropathologists summarize their frustration with lack of any specific neuropathological finding by calling schizophrenia the graveyard of neuropathologists. In 1970s and 80s, the development of brain imaging techniques like CT scans and MRI provided another view to examine schizophrenia in living beings. Initial or first few CT scans showed enlarged ventricles, and that has been replicated several times now. There are like few famous earlier imaging studies like twin study and then sequential study. They all confirmed that people who developed psychosis showed loss of gray matter in frontal lobe, and there was increase in ventricle size. And these changes were sometimes seen before the onset of clinical symptoms. This timing of onset of schizophrenia and the first change in the volume of the brain and the timings of processes like synaptic reorganization and pruning uh, led to this hypothesis that there is a faulty remodeling of gray matter, and combined with absence of gliosis, that led to the foundation of neurodevelopmental model of schizophrenia. So what do we understand from all the studies done so far at different scales? So let me, all right. Let me give you a summary of studies being done at different scales. And we hear them all the time, and we read them in different neuroscience journal. So on the left side, you see the studies at micro scale level, which can be done using microscope or other, or studied by using other methods, specifically focusing on subcellular structure and function. So neurotransmitters, genetics and epigenetics, microscopic changes in the cell structure, and inflammation or immune dysregulation, which we see on examination of body fluids. And on the right side, you see the macro scale studies, basically the neuroimaging and electrophysiology studies being done in the living human beings. So let's go through each of them and see how they inform each other, and how they are findings from the one scale is dependent on the findings from the other scale. All right. So first of all, talking about the genetics, the Psychiatric Genomic Consortium have looked at the thousands of patients, and they looked at the whole genomes of the thousands of patients and healthy controls, and found out there are 108 genetic loci which are associated with the risk of schizophrenia. And most of these proteins are in the known coding regions of the gene. So they do not code for any structural proteins, but or they're also called the junk DNAs, but they are involved in the regulations of processes. And these processes can be neurotransmission or neurodevelopment and other regulatory functions. And we'll go through them. As we go through the other processes, we'll see what how the genes affect those processes as well. Next, with modern microscopic techniques and these 3D evaluation or assessment techniques, uh, there were some structural changes noticed in the brain cells, especially the dendritic spine density. So each neurons has dendrites, and dendrites have the spines, and those spines are the means of communications with other dendrites. Uh, and there is a normal developmental process called pruning, which happens during late adolescence or early adulthood. And what happens there is that the excessive dendrites, or the dendrites which are not required for the optimum functioning of the brain, are removed. And it has a genetic basis. So the genes control this process, and this process leads to the optimization of the functioning of brain. And we lose a lot of dendritic spines. But what is noticed in the schizophrenia brains is that the schizophrenia, especially in the frontal and prefrontal cortex, the dendrites have fewer spines than the healthy controls. And therefore, the cells have the smaller size, and they need small space. And this small cell size and lesser dendritic spinal density leads to tight packaging of these cells in a smaller space, and that leads to the ventricular enlargement. So it's not the neurodegeneration or loss of cells, but it's the small cells and the lesser number of spines that leads to the smaller volume required for those cells. And this led to this another hypothesis called reduced neuropil hypothesis. So overall, the number of neural cells is the same as healthy control, but they are less efficient in their communications. And as we see in the neuroimaging studies, that there are deficiencies in the information processing, and that's global and widely prevalent across the brain, especially the connections from frontal lobe to other cortical areas and subcortical structures. And it's possible that that lack of or loss of dendritic spinal density is the reason for those faulty information processing. Uh, Inflammation or immune dysregulation plays important role. There is one hypothesis which suggests that schizophrenia can be an inflammatory process. And there are many risk factors which for schizophrenia, including genes which promote neuroinflammation. And what we notice on postmortem and clinical studies that there are increase in pro-inflammatory markers in patients with schizophrenia. And also there are elevated level of cytokines. There are a number of studies and also meta-analysis telling us that those cytokines are elevated in schizophrenia, especially in the acute episodes of or acute exacerbations of psychotic symptoms. Uh, Along with that, there are some longitudinal PET scanning studies which show that there is increased expression of a translocator protein, which is a marker of the pro-inflammatory microglia. And this increased expression of translocator protein may gives us information about the inflammatory process going on in the living beings, and that may lead to gray matter loss. Even though this protein is not a reliable marker, but it's a good enough marker to examine the in vivo neuroinflammation. Along with that, we know that there are several anti-inflammatory treatments which have been proposed for schizophrenia. For example, minocycline, fatty acids, COX-2 inhibitors like celecoxib and others. And they somehow modulate this immune process, leading to the improvement in symptoms. But like I said, what is most noticeable is that this inflammatory process is seen in only about 30 to 40% or 35 to 40% of schizophrenia uh, population. And there are some studies which suggest that these inflammatory markers can only be seen in acute uh, episodes. So that gives us to few theories such as that that provide additional evidence for theories that schizophrenia is a heterogeneous disorder and is composed of several syndromes, and it's not a one syndrome. So it's possible that inflammatory or inflammation associated schizophrenia may be one of the disorders. Now, neurotransmitters, which is the most studied and most widely used explanation for schizophrenia symptoms and the treatments. We'll come back to that in a little bit, but dopamine, glutamate or NMDA, and GABA receptors or GABA neurotransmitters are most commonly implicated. I will come back to these in a little bit, but there are enough evidence from the neuroimaging point of view, specifically MR spectroscopy, that all these neurotransmitters are somehow implicated. Now, these are the micro scale studies. At macro scale, what we notice is that electrophysiological or EEG and ERP studies show that there is altered mismatch negativity, which is like when we present an oddball sequence to schizophrenia patients, their reactions to odd stimuli, visual or auditory, is different from the healthy controls. Then pre-pulse inhibition, which is again a marker of decrease in habituation. Each sensory stimuli appears novel to schizophrenia people, and that may contribute to the symptoms of altered perception. And gamma band oscillations, which is again probably a marker of the loss or reduced functions of GABA receptors or GABAergic neurons in the cerebral cortex. Uh, so these findings on the EEG and ERP can be explained from the neurotransmitter alterations, which can again be seen visually by the neuroimaging methods. So what do we see on the neuroimaging? And let's go to that. So like I said, structural or CT scan studies of schizophrenia have repeatedly shown that there is a loss of gray matter and enlarged ventricles, and which is explained from the synaptic pruning hypothesis that there is genetic control of synaptic pruning, which is altered in schizophrenia, and that leads to loss of gray matter and uh, not exactly loss of gray matter, but uh, reduced size of the cortex and cortical folds and enlarged ventricles. In diffusion weighting studies, when we look at white matters, there is significant reduction in the connectivity between the frontal and temporal lobes, more on left side for some reasons. And there's increased extracellular free water, which may be marker of neuroinflammation again. On functional studies, based on the cognitive dysfunction we see in schizophrenia, including working memory and difficulty with facial affect, these tasks, when studied in the fMRI, are they provide significant information about how the schizophrenia patients show different activation on fMRI than the healthy controls. And in resting state fMRI, there is excessive activity in the hippocampus. And if we look at the overall brain organization from graph theory, there are changes in the way the brain is organized. So it's there is reduced clustering and small worldness, which renders brain less efficient in information processing, and it's more susceptible to targeted attacks from inflammation or any changes in the structure of those neural pathways involved in these. And finally, the molecular imaging, especially the PET and SPECT scans and MR spectroscopy, they again provide evidence for the neurotransmitter dysfunction, especially the dopamine and glutamate. And there is the uh, most widely reported findings from the MR spectroscopy is that there is excess of dopamine and glutamate in the thalamus, the subcortical structures. And I'm going to come back to that when we are going to talk about this glutamate hypothesis of schizophrenia and see why there is too much of glutamate there. Uh, so overall, what we see is that micro scale and macro scale studies do provide supporting evidence for changes at different scales. And it's very important to understand the schizophrenia uh, from the combination of these two scales and see how they are dependent on each other. Now let's go to the neurotransmitter hypothesis. And the most commonly used explanation for schizophrenia has been the dopamine dysfunction in schizophrenia, and that's based on the antidopaminergic medications we use. But there are some issues with dopaminergic hypothesis. One of them is that when we use dopamine agonists, they induce only positive symptoms and have no effects on the negative and cognitive symptoms as seen in the uh, schizophrenia. And also the antidopamine or antidopaminergic drugs or medications we use, they have effects only on positive symptoms. So Dopamine model of schizophrenia may not be the most appropriate explanation for the symptoms of schizophrenia, while NMDA or glutamate explains most of those findings. For example, effects of ketamine and PCP is very similar to symptoms seen in schizophrenia. They induce not only positive but negative and cognitive symptoms, and not only in the people who have history of schizophrenia but also healthy individuals. So ketamine uh, administration in healthy individuals can lead to slight decline in the working memory tasks or facial affect recognition, which is similar to what we see in schizophrenia. There are genetic models of rodents where NMDA receptors are altered, and that leads to function similar to uh, schizophrenia models of the animals. Then mismatch negativity, which is again a marker of glutamate functioning of the brain, is seen in schizophrenia. And when the postpartum brains, I'm sorry, of schizophrenia people were looked at, they show the changes which support this NMDA hypothesis. So what glutamate or NMDA does to brain is that uh, it's a principal excitatory neurotransmitter. And NMDA and GABA are the excitatory inhibitory this uh, it's a stable state of brain where there is a balance between excitatory glutamate and inhibitory GABA. And most of the glutamatergic neurons are found in the cortex or the cortical layer 2 and 3, where you see these large pyramidal cells. And these large pyramidal cells provide projections to other cortical areas as well as thalamus or subcortical structures. Uh, so the NMDA antagonists like ketamine and PCP uh, should, if we take it like as it is or simply understanding, NMDA antagonists are going to inhibit the NMDA receptors and should lower the functioning or uh, lower the glutamate release from pyramidal cells endings. But what happens is exactly opposite, is that there is hyperglutamatergic state in the thalamus and other cortical areas. And the most uh, correct explanation for that is that the functions of these large pyramidal cells, which release glutamate in the cerebral cortex, is dependent on the GABA interneurons, which are smaller neurons seen in close proximity to these large pyramidal cells. These GABA interneurons have NMDA receptors. And remember, the pyramidal cells, they also have NMDA as well as EMPA receptors. So their functions is not only dependent on the NMDA receptors. But these GABA interneurons, uh, when they are blocked by the PCP or ketamine, which are NMDA antagonists, leads to the disinhibition of these pyramidal cells. And this disinhibition leads to the excess of glutamate in the thalamus and other cortical areas. And this excess of glutamate can be observed using EEG in terms of mismatch negativity and other similar paradigms. Excess of glutamate in thalamus can be observed by MR spectroscopy. And then fMRI. Uh, fMRI changes can be seen, which are results of the glutamate excess. And one of them is that from cortex uh, via thalamus, these glutamatergic neurons also project to hippocampus, and there is excess of excessive excitation in the hippocampus, and which is which can be observed by seeing increased blood flow in the CA1 subfield of hippocampus on fMRI. And this hippocampus overactivity leads to excess of dopamine release from ventral tegmental area. And if we look at normal memory functions, we can understand this relationship better. So all right, well, I should have moved these slides while I was talking, but anyways. So memory, when we have the memory which is associated with novelty or a drama, we retain them better than just the routine information we get from the environment. And one of the reasons for that is that hippocampus stimulate the ventral tegmental area, and there are emotions associated with those memories, and they are consolidated stronger than the other memories. So we know that there is relationship between hippocampus and ventral tegmental area, but the excess of glutamate also lead to this excess of dopamine release from ventral tegmental area through hippocampus. And this excess of dopamine leads to the positive symptoms. So uh, there are strong evidences now that the dopamine abnormalities can be secondary to glutamatergic abnormalities. And excess of glutamine or glutamate in thalamus has been correlated with the severity of symptoms and cognitive impairment in schizophrenia. So what we see here is that all these neurotransmitter alterations do lead to these changes at the macro scale level, which can be observed with EEG, fMRI, and MR spectroscopy. And we can also correlate these neurotransmitter changes and the neuroimaging changes with the clinical symptoms, which are noticed on the clinical assessments. Another important hypothesis is neuroinflammation. So infection during pregnancy leads to this immune activation, and that prime the microglia in the fetus. Then other environmental changes like pollutions and heavy metals and toxins or early life stress. And then there is this gut-brain axis, that altered microbiome or altered bacterial composition in the gut can lead to this chronic low-grade inflammation or inflammatory changes. And these chronic low-grade inflammatory changes can have effects on the brain, causing this very chronic and low-grade neuroinflammation. And there are enough evidence to support this hypothesis based on the blood levels of cytokines and activation of microglia. So microglia, when they're assessed using the translocator protein, which is a marker of activated microglia. Uh, uh, Again, I mean, this activation of microglia or loss of balance in the microglial functions can affect the structure in terms of altered synaptic plasticity or altered synaptic formations. And we have seen that under microscope, neuronal cells show less spinal density in schizophrenia. So microglia may be the culprit in that process. And the anti-inflammatory medications trial, again, provide the support to this hypothesis of neuroinflammation. So overall, what we see is that there are the structural changes or neuroanatomical changes in the brain, which have genetic basis and can be associated with symptoms or clinical profile. Then there is neurotransmitter hypothesis, and then this immune dysregulation. So these are the major hypotheses which provide evidence for or evidence for pathophysiology or etiology in schizophrenia, and we see that how they are codependent on each other or interdependent. So let's take a few examples of what we exactly see in the at the different scales. So this is a micro scale study of the pyramidal neurons, and if you pay attention, you see that in schizophrenia group, there is significant uh, there is significantly low density of neurons in this the third spinal layer, and these changes are mostly seen in the frontal and temporal neocortex. And if we look at the brain organization from the graph theory point of view at the macro scale, what we see is that in schizophrenia, the modules are not densely connected or not as densely connected as in normal individuals. So the top ones are normal or healthy controls, and the lower ones are schizophrenia. So uh, thereby there is the edges within the modules, say there are 76% and 67%, and edges between modules is 33% while it's 24%. So what it does is that it reduces the efficiency of schizophrenia overall, and we can say that this disruption in the organization at the graph theory level or graph theory scale make it less efficient. Now, all these studies or most of these studies are done by either the neuroimaging researchers who are focused only on the neuroimaging studies, or pathologists who are looking the cells under microscope, or immunologists who are looking at only the immune markers. So when we look at their correlations or their interdependence, it's mostly circumstantial. There are very few studies done simultaneously at different scales. And that's the point of this micro scale or multi-scale neuroscience, which is coming up, or like a relatively new field in the neuroscience, that instead of getting evidences from different studies and then uh, making assumptions, if we combine the studies at different scale, that will be more useful in understanding of pathophysiology. And in this study, what you see, especially in this, the D point, it's that there is significant association between cortical expression profile of the schizophrenia risk genes and pattern of macro scale disconnectivity on diffusion MRI. So there is a strong correlation, and uh, that provides the evidence for the interrelationship between the genes and the findings on the diffusion MRI or white matter connections. Here's another interesting observation. Here, what you see is that the changes in the macro scale connectivity is also highly correlated with the spinal density changes in the cortex. So when we say that the loss of spinal density or loss of the structure in the cortical neurons uh, have implications to what we observe under the MRI is true based on these findings, that the white matter connections are in fact dependent on the spinal density in the cerebral cortex. Now, so eventually the goal of all these neuroscience studies is to understand the pathophysiology, and improve the clinical care of the patients. And the way this neuroscience can contribute to the clinical care is by providing us a biomarker which can be used to identify the disease before clinical expression, or can differentiate it from the other non-related psychiatric disorder, and or inform the treatments. So uh, why don't we, I mean, even in spite of all these understanding at different scales, why don't we have the biomarker yet, which can provide us information? And there are several challenges towards that. One of them is that there is this inherent tensions between the DSM diagnosis, which is based on the clinical symptoms, and the like the what we observe in uh, the neuroimaging and electrophysiology studies, that the phenotype from these objective studies. So there is a group called BSNP, or Bipolar Schizophrenia Network on Intermediate Phenotype Initiative, and they have published a few interesting research over past few years. And so this is one of their examples. So those circles are the show the DSM diagnosis. There is schizophrenia and the bipolar, and this overlapping is schizoaffective disorder. And when they try to differentiate this whole group of cohort from the objective neuroimaging and electrophysiology findings only, they found they didn't find much overlap. So what they see is that there are three different phenotypes, and and that covers all three different disorders. So it's possible that when we are using these clinical diagnoses based on DSM to find a biomarker, we are looking at or we are like making mistake from the beginning of this hypothesis, or in I mean, the whole experiment is flawed when we are looking at this very heterogeneous group of people and trying to find a biomarker for that. So this approach of this BSNP initiative, or the RDoC criteria, the research domain criteria paradigms from DSM, can provide better answers to our quest for biomarkers for schizophrenia. So what we expect is that this kind of research will eventually provide us objective markers for a biomarker. And what biomarker means is that we should have a objective marker which can detect a particular psychiatric disorder by at least 80% sensitivity and 80% specificity. Um, and it seems like, based on the different scale studies, that neuroimaging studies, which may not be the primary abnormality in schizophrenia and may be only the consequences of genetic, neurodevelopmental, and neurotransmitter changes, can provide us important markers and can be used in living beings for the diagnostic classification, or even identification before clinical expression, or can inform treatment, and the treatments based on those specific pathophysiological mechanism can provide to can lead to the better outcomes in our patients. So that was it from me for today. I hope that I contributed a little bit to your understanding of schizophrenia and the neuroscience at different scales. And we can discuss few questions from the audience now. Hello, Dr. Bhaskar.
So I'll just try to get him, I think.
Okay.
Hello.
Yes, Dr. Bhaskar.
Yes. Sorry for getting disconnected. Actually, wonderful session. Um, I would like to see what are the questions that have been asked by our audience. Amita, please send me the questions.
I've sent it on your WhatsApp, so can you just please check?
Yes. I have sent them. Okay. There are actually 5 questions.
Okay.
Uh, One is from Dr. Paresa. Suicidal brain of be it schizophrenia, be it bipolar, be it major depression, are they the same or there are structural differences?
Well, there are structural differences, as we know that schizophrenia is associated with the structural changes at microscopic level in form of like the loss of spinal density and also the loss of gray matter at neuroimaging level, which is not seen in bipolar or depression as much. So there are definitely the difference in structures at different levels too.
And I would like to add something of my own. I would like to add something of my own that
Yeah,
schizophrenia, bipolar, and major depression, these are just name of some social concepts. These are not diseases. These are some symptoms which we categorize
exactly.
as diseases. So
Exactly. Yeah.
standing there today, we are diagnosing the patient as having major depressive symptoms. Tomorrow, that same patient would develop obsessive compulsive symptoms. Day after tomorrow, there might be some manic symptoms, and a few days down the line, that same patient might develop psychotic symptoms. So, the on longitudinal view, if we ever employ, we don't employ language, we employ actually cross-sectional view while seeing a patient. If we start seeing longitudinally, we found that this schizophrenia, bipolar, they are not really the endophenotypes we are looking for. The personalities, they are more of a behavioral and cognitive endophenotype. And if we go deeper, then the diseases gets apparent. And standing there, so a patient of depression who has underlying synaptic dysfunction will show same feature as a patient of psychosis who has synaptic dysfunction. But if there is a patient who has neuroimmune disturbance along with synaptic dysfunction and is showing suicide, depression today, then that patient would not be same as our previous patient. So there would be structural differences both in systems view as well as in individual, microscopic, and molecular.
Absolutely. And again, I mean the uh, most important learning from studies so far is that, especially with this last BSNPs example, is that if you look at these disorders from the the RDoc point of view or from the phenotypic point of view, which are developed from objective assessment, that will provide us more useful information than just looking at the DSM related disorder diagnosis and their neuroimaging or neuroanatomical findings.
Yes, exactly.
Yeah.
Now, there is another question of Dr. Paresa, that deviation of psychological formulation based on psychoanalysis helps resistant chronic schizophrenia recover much better than treating only with pharmacotherapy. What is your take? Is there any study that assessed usefulness of psychoanalytic treatment along with pharmacotherapy to treat schizophrenia? So he wants to know whether psychoanalysis plus pharmacotherapy has any role in chronic schizophrenia.
Well, I'm not aware of any such study. I don't know if you know something about this.
There are few studies, but they are unstructured.
Okay.
So, as far as I remember, in Scandinavian countries, they are trying to combine neuropsychoanalysis and pharmacotherapy in schizophrenia. But so far nothing. So far nothing.
Okay.
Okay. Now, another question is Dr. B. Diagnostic classification versus prognostic analysis. What's the difference?
Diagnostic classification
versus prognostic analysis. What's the difference?
Well, I'm not sure what was the intention of this question,
Yes.
but
Same.
But again, I mean, if you look
Very
from the
question.
Yeah, but if you look from the the biomarker point of view, there can be different biomarkers for diagnostic classification and also to assess the prognosis or to estimate the prognosis, and we have those examples in the other medical illnesses too.
There is another question that how can disproportionate anxiety symptoms not amenable to other treatment be only sign of early psychosis?
I didn't really get it.
It is asked by Dr. Srivastava that can disproportionate anxiety which is not responding to other treatment, can this be the symptom of early psychosis? Yes. Sometimes yes, most of the times no.
Okay.
Actually, this is a question which should not be asked in this topic because this is not a question of this.
Right. It's not very related to the topic of discussion. Yeah.
Yes. And Dr. Ramesh Babu has asked another question, that multi-scale entropy analysis and its effect to understand the psychopathology of schizophrenia.
Yeah, I mean that's very important, and it's been ongoing right now. I mean, there have been a lot of emphasis on that in the current schizophrenia research. Yeah.
And along with that, he also added an extension that whether this would help realizing that gender difference in schizophrenia treatment and gender difference in schizophrenia symptoms. Whether the entropy analysis would be good enough to answer that.
Yes, so there are a lot of studies being done in that subject area, especially because we know that the genes and environment are important uh, predisposing factors for the clinical expression of schizophrenia, and like all these genetic vulnerabilities in schizophrenia individuals uh, are like they leads to only vulnerabilities, and the environmental factors usually trigger the expression of disease, right? So it's possible because the men and women go through different experiences and different changes in their during their development and during their like course of life. So obviously they are subjected to different kind of stress, both internal and as well as external. So it makes sense that there may be gender differences in how this whole pathophysiology work in the men and women differently. Uh, but I mean that's my assumption based on how the environment affects the expression of disease.
No, along with that, there is also now evidence that all the proteins of body works differently in both sexes. So
Hmm.
various proteins, various transporter molecules, various enzymes, they work differently in male and female. So that probably is one of the reasons that there is difference in drug response as well as symptom expression in male and female. But we don't know the exact molecules involved in schizophrenia. But because again, schizophrenia is not a disease, it's a symptom. So
Exactly, yeah.
unless we get the pathophysiology, it's very hard. This is the
Yeah.
last question. This is the last question. Thank you very much for coming here and doing this wonderful lecture.
Thank
It's
you,
an
thank you,
honor
and
to have you.
I appreciate this opportunity. Thank you so much.
Mhm.
Thank you, everybody.
Thank
Dr. Bhaskar,
you.
so you'll have to
Yes.
announce the best question.
The best question again today would be Dr. Ramesh Babu. Dr. Ramesh Babu. Hello. Yes. Hello.
Yes, sir. Yes, sir.
Thank you all. Thank you all for joining us today. We hope to see you again on coming Tuesday. Please block your date from 16th March 2021, same time from 8 p.m. to 9:30 p.m. Topic for our next webinar is Delirium, a medical emergency with psychiatric manifestations by Dr. Jigishu Divatia and Dr. Sandeep Grover. Also, we would, based on many requests that we have received, we would like to inform you all that all our previous webinar archives are now available on our YouTube channel, Banai India. Do send us your feedback and comments, if any, on info@banai.org.in. Till then, stay safe. Good night.